Hello, I'm Larisa Rotariu. I'm a medical oncology resident from Iași, Romania, and a PhD student in immuno-oncology. I accepted Ilias' invitation, and I'm really happy to be here with you today to talk about NGS or PCR for mutation detection in non-small cell lung cancer, an agnostic approach in patients who have received standard treatment. I have no disclosures. About NGS, it is a technology that have revolutionized genome, expanding our knowledge of genome structure, function, and dynamics. It has enabled extensive research and also allowed scientists to explore the complexities of genetic informations. This progress has paved the way for significant breakthrough in the field of DNA sequencing, leading to the emergence of three generations of sequencing technology. The first one was represented by Sanger sequencing, um, providing the foundation for the DNA sequencing. The second one introduced the massively parallel sequencing with platforms such as Illumina and Ion Torrent. And the third one, the current one, include PacBio and Nanopore. So how does NGS really work? There are four big steps. The first one, the DNA fragmentations, it is used to break the targeted DNA into many short segments. It can use mechanical methods or enzymatic digestion. We have two assays. The first one is hybridization capture, which uses a mechanical method, and the applicant assay, which uses PCR. The second one is the library preparation, which is a process by which DNA segments are modified so that each DNA sample can have a sample specific index, and it allows the sequencing adapters to be added to the DNA segments. The third one, the sequencing, which represents the massive parallel sequencing performed using an NGS sequencer. Different sequencers have different sequencing matrices. Like, for example, Illuma NGS sequencer uses flow cells, and the Ion Torrent sequencer uses sequencing SIPs, but they all have the same goal to allow massive parallel sequencing of all the DNA segments at the same time. And the last step, the analysis, which is the process involving the base calling, real alignment, variant identification, and variant annotation. But why do we really need NGS? We can use it for the whole genome sequencing, which is costlier, and we can detect deletions and duplications. We can use it for exome sequencing. It is a reasonable approach for some clinical situations because over 85% of known disease causing mutation are found in exomes. But the main disadvantage is that this exome sequencing could potentially miss a pathogenic variant in a non-coding region of the genome, and we have targeted gene panels. In oncology, we use it for the hereditary breast and ovarian cancer and the hereditary colon cancer. For interpretation, we have five possibilities from pathogenic to VUS. About PCR, PCR underlines many molecular biology and molecular genetics techniques. In just a few hours, PCR can create a million copies of a single DNA or RNA molecule, providing sufficient material for clinical and research applications. There are many times as listed, and there are four steps. After the reaction mixture is assembled, the three steps are repeated from 30 to 40 times. The first one, denaturing, is where the mixture is heated to 95 grades to denature the double-stranded template DNA into single strands. The annealing one, where the mixture is cooled and the thermostable DNA polymerase can bind to the three end of the primers. And the last one, elongation, where the temperature is raised again to a temperature at which the thermostable DNA polymerase can initiate this process. It continues for about a minute, and it is terminated by raising the temperature again to 95 grades to restart the cycle. There are advantages and disadvantages. An advantage would be speed. It takes about an hour or maybe 2 to 4 hours if it's a large scale. The detection limit, it can amplify DNA from a single cell or a single molecule, and the low cost, but with some disadvantages that it cannot identify new DNA variants or new microorganisms, and there are challenges in identifying rare sequences. So we have the non-small cell with oncogene addiction map. Oncogene addiction tends to make driver mutations good biomarkers for selecting patients. Screening for them has become increasingly standard part of the diagnostic workup in non-small cell lung cancer. Besides the immunohistochemistry, which is the only way to test PD-L1 in all non-small cell lung cancers, regardless the histology, and maybe FISH used for some mutations, nowadays we have these two techniques, NGS and PCR, to test specific alterations. But how do we choose? This is a table with the most common driver mutation. We can see the high sensitivity in uh, in some cases for both PCR and for NGS in tissue and in liquid biopsy. The first one, EGFR mutations, the most common one are as listed. We can use both NGS and PCR. In my institute, we perform mostly mutant allele-specific real-time PCR to detect them. However, there are studies which says that several uncommon mutations lying outside the coverage area of the pre-designed primers may be missed, compromising the patient's clinical outcome. For example, a group of Taiwan did a study on a group of stage 4 non-small cell lung cancers. They received the standard molecule, which is RT-PCR, and then they undergo NGS in patients, EGFR ALK double wild type patients to identify potentially actionable mutations. The results from a group of 87 patients, a total of 24 had driver mutations detected by NGS, and in 7 of them there were found EGFR mutations, and most of them, 6 out of 7, were exon 20 insertions, followed by uncommon exon 90 deletions. So the main method may be PCR, but maybe in some cases NGS is better. About ALK, we can use immunohistochemistry for screening. There can be false positive and false negative results. Fish can also provide both false positive and false negative, and we need the presence of split signals in more or less than 50 of the cells to assess this mutation, this rearrangement. In this case of ALK, RT, PCR, and NGS, they are not preferred. RT-PCR provides mainly false negative results. The limitation is very specific, resides in its ability to detect only known translocation and more specifically those that the assay was designed to detect. Maybe NGS would be a better option because it covers almost all variants described to date, including partners other than MLL4. About ROS, we can use immunohistochemistry for screening. However, in positive cases, the concordance rate can be as low as 79%, indicating the necessity for a confirmation test. We can use here PCR, quantitative PCR, to detect reported fusions, but NGS in this case is preferred. We can see here the advantages and limitations for both techniques. PCR has a short turnaround time, while NGS has a longer turnaround time. They both have high specificity and sensitivity, but ESMO recommends NGS for this type of rearrangements. About red, Even if fish technology is the gold standard, NCCN and the Chinese Society of Clinical Oncology recommend NGS for this type of mutation, because and the RT-PCR in this case has a big rate of false negative results. For NTRK, we use immunohistochemistry screening even in my institute. But ESMO recommends NGS for confirmation. In my institute, we do RT-PCR because we do not have an NGS, and maybe in the future, more development of RT-PCR kits would be an option. So there are a lot of other tumors like those listed, and in these cases, sometimes better to use NGS if we do not know what we want to find, but if we are specifically sure about the mutation, we can use PCR if we have the primer. So, an agnostic approach in patients who have received standard treatment. When I saw this title, my first question was, why do we have to treat our oncogene addicted non-small cell lung cancer with standard treatment? There are two answers. Maybe we do not know that the patient has a driver mutation, and in this case, I want to emphasize that it is really important to test them with tests like NGS or PCR if it's possible, and we have to have a good tissue or good liquid biopsy. Or maybe we know that he has driver mutation, but we do not have treatment in my country. There are a lot of driver mutation without a reimbursed treatment. So we have to address them for a clinical trial. For this, to emphasize my main idea, I will present you a clinical case, a non-small cell lung cancer stage 4, EGFR wild type, ALK negative with the expression of PD-L1 of 30%, case, a male, a smoker, diagnosed in January 2023. We started standard treatment with carboplatin, pemetrexed, and pembrolizumab. And in June, after fourth cycle, we had a pseudo progressive disease. We decided to continue the protocol, but after 3 more cycles of PAM and PAM, we we had a clearly progressive disease. And we started the second line with docetaxel, but in December 2023, we had again a progressive disease with new target lesions and the new anterior left thoracic nodule from which we took a biopsy. I'm sorry that the results are in Romanian. I couldn't translate them. But here, first, they performed NGS, and where I pointed is what I pointed is that they say the tissue was not good enough for driver mutation testing, so they performed PCR, and they found the mutation in the exon 19 for EGFR. So we started in February 2024 osimertinib, but unfortunately it was too late because he had progressed, he had brain mets, and after 2 months of treatment, we lost him. To sum up, we have to test all the patients with non-small lung cancer, mostly adenocarcinoma, for driver mutation. In my institute, after progression, we try to perform a new biopsy and do NGS. Sometimes NGS is better for some cases, sometimes PCR is better, but you cannot have it all. It depends on the resources and the reimbursements. Okay, but what if we test and we find the mutation, but we do not have the treatment? My opinion, uh, we have to address the patient for a clinical trial. Thank you for listening.